This is Mrs. Palmer-Quay with the second video for Unit 12. In this video I will cover the first 6 goals or learning targets under Goal 2. So this is covering the immune system, its basic structure and function. There will be one more video that will cover some specific disorders. The function of the immune portion of the lymphatic system is to protect our body from invasion by different pathogenic microorganisms. So we have some examples over here in this illustration: viruses, bacteria, the toxins that bacteria produce, fungi, parasites, just the germs in general that we don't want to have in our body. The immune system protects us from the infection caused by these pathogens moving in to live in you. The immune system can also deactivate the toxins that are produced by the bacteria, and when it comes to your own body producing mutated cells that could potentially become cancerous, in many cases your immune system can destroy them before they get established. There are two specific layers of defense. We have the innate or non-specific defense, and then we have the specific defense. So we're going to start with the non-specific defense first, which gives us general protection against a variety of pathogens. And there are 5 different ways that we have this sort of general barrier to being infected and suffering from the attack of some foreign substance. The first way that we have a general defense is that we have a genetic resistance against certain diseases. So many viral diseases are very species specific. That's something about how the body of whatever particular species we're talking about makes protein. Remember, viruses are not alive and they have to take over a host cell. So there's something in how the host cell does its protein work that the viruses are not able to get established in that particular species. So we can get measles, we have to get a vaccination to prevent measles, but our dog cannot get measles. So we cannot spread certain diseases from ourselves to our dogs. There are a few things that cross species lines, depending upon what species of animal you're talking about, but in many cases they don't. For example, also the HIV virus came into the human world out of the monkey population, but monkeys are not sick with AIDS. Also, all living things have genetic variation, and so there are family lines that have a stronger resistance to certain bacterial infections. And this is something that's important to livestock producers, especially those in the sustainable agricultural field that are trying to move away from using a lot of medicinal chemicals and such in their animals, looking for family lines that seem to be genetically resistant. We've seen this in humans as well, but we're not breeding humans for profit, so nobody's selecting quite the same way in the human population. But it does exist. You may naturally have genetic resistance to some bacteria and would not get sick if you were infected with that bacteria. Another general defense are the physical barriers that we have. Our skin is a wall around our body. We're running into bacteria all the time. Every surface we touch, every person we touch is covered in bacteria, but that bacteria generally stays in the outside environment because we are covered in the armor of our skin. So this is our first line of defense. Not only your outside skin, but the mucous membranes that line all of those cavities that are open to the outside are designed to prevent entrance of pathogens, to keep the pathogens out. And the fact that we've got fluids moving in our body, our tears are washing out of our eyes, sweat is washing off our body, the digestive system fluids are moving through the urinary system, the urine is cleaning out the urethra out of the bladder. These are all things that continually wash pathogens away from the body as well. Then there are the chemical barriers that work with the physical barriers to keep the bad guys out. In these fluids that I mentioned, we have enzymes, or they're high acid fluids, which helps destroy these pathogens before infections can occur. So lysozyme is an enzyme that is present in saliva and in tears, which will inhibit bacterial growth in those two fluids. The fact that sweat is salty will also be enough to stop some bacteria. The environment, too salty of an environment keeps them from growing. We've got stomach acid that breaks down a lot of pathogens before they go any further than our stomach. And in fact, the ciliated cells in your respiratory tract are continually moving the mucus that lines your respiratory tract up. It doesn't move it down into your lungs, but it moves it up to the back of your throat so that you can swallow that mucus and deposit those bacteria and such that are caught in the mucus, deposit them into your stomach to be digested. And then the sebum that is put out by your skin also lowers the pH of your skin somewhat, so you're a little acidic on the outside of your body, and that will also inhibit bacterial growth. Some more specific proteins or defenses, chemical defenses, small proteins called defensins, which are secreted by leukocytes, white blood cells, and certain epithelial cells, also will break down the cell membranes in bacteria and fungal cells to keep them from surviving. Certain defensins also are involved in preventing viruses from entering host cells. Cells that have been infected by viruses are able to produce interferons, which interfere with future viruses being able to move into surrounding cells, and so that it slows down the overall viral reproductive cycle. And then there are a group of proteins known as the complement group. These proteins are found in the plasma, and they are involved in a number of defenses in our immune system. They're involved in encouraging inflammation and encouraging phagocytosis by your phagocytic cells, the macrophages and all. And they also can work together to create an opening in the bacteria, punch a hole in bacteria so that they're not able to survive. Here's a chart that covers some of the things I just said. A couple new things on this chart is emphasizes that epithelial cells, your skin cells, are joined together by tight junctions, so there's no gaps in between cells where anything could slide in. It also mentions here on the bottom of the chart that we have our own normal level of microorganisms, our normal microbiota, present both on the outside and the inside of our body, and they're taking up space. That's a part of the way that we do defend against invaders, is that there's no room for anybody to come into the neighborhood, because our body is well populated with healthy, safe, not dangerous bacteria both inside and out. Then we've got cells that are defensive cells that respond generally the same to any kind of attack. They're not specific, because we're still talking the non-specific defense here. So some of the defensive cells that are working in our bodies all the time, they're a type of lymphocyte called a natural killer cell, which are involved in destroying viruses by destroying the host cell that the virus is living in, or our own mutated cancer cells, by causing the cells to break apart by secreting a substance called a perforin that makes a perforation in the cell, a hole, punches a hole in the cell. That's something else that the complement proteins can do. They also create this ring, a perforin ring. Then we have the phagocytic cells that are going around. They are mobile, they are able to move throughout the body, and they are able to then engulf, to take in any small bits of bacteria or virus pieces that they run into as they're traveling around the body. So the phagocytic cells include our macrophages, the neutrophils, the eosinophils, and then the Langerhans cells in the skin, which are a dendritic cell that are fairly newly discovered. We don't know a lot about how they work, but these cells are all phagocytic. They're all able to engulf and destroy foreign material by taking it in and digesting it. And then other cells that we've heard about when we were talking about the white blood cells, the basophils and our mast cells, are able to secrete histamines and increase inflammation, and also secrete other chemicals that draw the phagocytic cells to the area of inflammation. Inflammation we'll talk about later, but inflammation can actually help stop infection because of the increase in heat and the way it is able to seal off the infected area until the white blood cells can take care of the problem. So moving on to inflammation, you know what it feels like to have something inflamed. There's basically 4 characteristics that are present. You will see redness and swelling. You'll be able to feel heat, and most likely you'll also be able to feel pain. So we have a picture here of somebody who has a boil, and it's exhibiting those 4 characteristics of inflammation. So this chart outlines what happens in inflammation and why you have those characteristics occur. So we start at the top. There's some kind of tissue injury that's allowing, there's either been a break in the integrity of the cells, or you've got an introduction of some foreign invader. And so immediately you get factors, chemicals that have been released by the injured tissue that are going to call leukocytes to the area, call white blood cells, and this is leukocytosis, is an increased number of white blood cells. And so these chemicals are going to be attractive and bringing more white blood cells to the area. Other chemicals that are released, histamine to cause inflammation, to make the blood capillaries more leaky so that more plasma goes into the interstitial space. The complement proteins are activated, prostaglandins, we talked about that in the endocrine system. These are all involved in encouraging vasodilation of your arterioles, so more blood is coming to the area, but also making the capillaries leakier, they're more permeable, and so that will increase the blood flow, and the capillaries will end up working its way down to more fluid being in the interstitial space, which then leads to swelling, which also can, as a side effect, sometimes lead to pain as a background. But bringing more blood to the area and bringing more just more fluid in that whole place means you've got an increase in oxygen and nutrients, so that the white blood cells that have been migrating into the injured area will have nutrients to power their activity as they are busy phagocytizing, I'm not sure if there's a word there, any kind of dead tissue or any foreign invaders they see. They're doing the cleanup crew. The increase in blood is what's going to cause a red appearance because there's more red blood cell coming into the area. And then also there are chemicals released by the injured tissue that will cause an increase temperature. It'll raise the set point in the hypothalamus of your body temperature. And so you get a fever, which if the infection is bad enough, you get a full body fever. If not, you just get at least a local area of heat. That increased temperature, your white blood cells work better if it's slightly warmer, and it also tends to be the bacterial invaders are less able to function, so they don't do quite so well if the temperature rises a little bit, but your white blood cells are more active and better able to phagocytize the infective agents, and so they work. This adding extra heat is to the benefit of your healing and to the detriment of the infectious agent. Fever, beyond just a localized bit of heat, fever refers to the whole body temperature going up, and as I said, there are chemicals released by the area of infection or injury called pyrogens, and they work in the hypothalamus to change your set point, so they increase the thermostat so that the body will reset itself to a slightly higher body temperature. So as I already mentioned, this will increase the activity of your phagocytic cells, and it inhibits the bacterial action. One of the ways that it slows down bacterial and fungal growth is that when your body is at a slightly higher temperature, the body sequesters iron. It takes iron out of the blood and kind of hides it away in the spleen and the liver. And the iron is not available to bacterial and fungal cells, and so that will slow down their reproductive process because both of them are very dependent on iron. And so it's kind of a indirect way fever will help, but it can be very effective. So that covers our non-specific responses. Let's look now at the specific responses, the adaptive responses. These are our body is able to change and adjust, that we have an immunity which develops after an exposure to a pathogen because our body is designed to recognize and respond to the threat. It is able to create memory cells so that the second response will be even faster. There are two different ways that our body creates this immunity. There's a cell-mediated response that's involving the T cells and the macrophages. And then there's the antibody-mediated response, which is involving primarily our B cells with a little bit of help from the T cells and the macrophages. This one is also known as humoral immunity because it's taking place primarily in the fluids, in the blood and the interstitial fluid, whereas the cell-mediated immunity takes place by interaction between T cells and infected cells. B cells and T cells were mentioned as leukocytes or lymphocytes when we were talking about leukocytes. So leukocytes are white blood cells. B cells and T cells are lymphocytes. They are made in the bone marrow like all blood cells. A stem cell is a cell that is undifferentiated. So there is the stem cell that can become many types of blood cells that are being made by the bone marrow. And then, some of them go on to become red blood cells. But those that become white blood cells, some of them differentiate into these B cells and T cells. The B cell remains in the bone marrow and then goes through a number of steps to become sort of a working B cell. And we're not going to really worry about these different types of B cells. But just know that the B cells remain in the bone marrow until they are pretty much fully developed, whereas the T cells travel out of the bone marrow and they take up residence in the thymus, and they undergo the end of their differentiation in the thymus. And if you remember from what we were talking about earlier, the thymus is really active in the years before puberty, so that your T cells are all being developed and specialized while you are still a child. We now know there's many different types of T cells, basically because we've had more sophisticated techniques in the laboratory for differentiating between them, but we're going to just talk in general what T cells and B cells do. So this chart's a little more specific than what you need to know. B cells and T cells need to be activated before they can do their job in their immune response, and they're activated because of antigens. We talked about antigens in reference to the blood system and the A and B and O types in blood. Antigens, if you remember, are molecules that are found on the surface. They're found on the cell surface, and they are some kind of marker. So a little picture down here gives you just sort of an artist's idea of what the antigens look like, sticking out from the cell surface. They can be some type of protein or a polysaccharide, a carbohydrate, or a glycoprotein, which is a carbohydrate plus a protein, or a glycolipid, which would be a carbohydrate plus a lipid. And so your own body, somehow before you were born, your body keeps a running inventory of what all your cells look like in you. And so your immune cells, your B cells and your T cells and your macrophages and all, know what cells are self, what cells belong to you. And so as a response, your body then will only look for those things that are not you, that are non-self. And it is based on these antigens that are found on the surface, so that as a B cell or a T cell or the other immune cells are sort of going around and bump into cells, if they find an antigen that matches the kind of the code on their surface that says this is a self cell, this is one of me, then it will not attack it. But if it bumps into a cell and it doesn't have sort of the recognition, the secret handshake, the right kind of antigen on the surface, then it will be identified as a foreign invader and be attacked. In the cell-mediated response, it starts out with a macrophage, or a couple other cell types are involved, but we'll just talk about the macrophage, and it eats a pathogen. It phagocytizes a pathogen, digests it, and then takes those antigens and sticks them out on the cell surface by this is the identifying protein, the major histocompatibility complex, the specific part of that outside cell that says this cell belongs to me, this is a self cell. And so this antigen is put out there, held near the MHC. And a helper T cell, a specific type of T cell, when it bumps into one of these macrophages that is presenting a foreign antigen on its surface by the MHC, they are activated and are sort of turned on so that they can be involved in this immune response. once your helper T cells are turned on because an antigen has been presented to them, it's like the macrophage is running up and saying, look, look, there's something you have to take care of here. So then it undergoes mitosis many times to form two different specific types of special T cells, memory T cells that are going to remember this antigen, and cytotoxic T cells. Cyto meaning cell, toxic meaning kill. These are T cells that are going to go out and defend the body. In this whole process, we have particular proteins called cytokines that are secreted. One specific kind is the interleukins. There's interleukin 1 and interleukin 2 especially, that help speed along this process of moving from an activated helper T cell to the cytotoxic T cells with some memory T cells. So the cytotoxic T cells then destroy foreign cells or cells that have been infected by a virus by actually touching the cell and inserting a kiss of death, doing something that will cause that cell to fall apart. Cytotoxic T cells are also releasing cytokines which attract more macrophages to clean up the mess. After they blow up a infected cell, then somebody has to vacuum up the debris. So here's a picture that sort of is trying to show that. Here you have a macrophage. Here we've got, this is just your T cell, what it looks like. Here's a macrophage that's busy engulfing a bit of foreign matter, and so it presents the antigen from that foreign matter here on the surface of the macrophage. This is run into by the helper T cells, so they connect, and it recognizes that here is the antigen it needs to respond to. It produces these little circles are the cytokines that are helping the maturation process, and so then it turns into the specific T cell. Changes shape a little bit here to give you a picture. Some of them, is going to be reproducing. You're putting out lots and lots of clones of the activated cell here, and so we've got our cytotoxic T cell down here that will, when it runs into that infected cell presenting that same antigen, will then be able to destroy the cell and take care of that particular invader. The second type of specific response is the antibody-mediated response. And this is where antibodies are made and they bind to the antigens and cause things to happen. The antibody-mediated response starts with a B cell. So now we're moving on to the B cells. They need to be activated again. All these immune systems don't work unless the cell is activated. So a foreign antigen is presented to the B cell either by a helper T cell, most often by a helper T cell, but it also can be directly encountered by the B cell. In some cases, the B cell can respond to the antigen directly. The activated B cell then divides many times to form memory B cells again and plasma cells. And plasma cells are the ones that actually make the antibodies. Antibodies attach themselves to this foreign antigen or to the cells that are carrying the foreign antigen, or perhaps it's the toxin that has the foreign antigen on it. And by doing so, they mark those cells, and that makes it easier for them to be destroyed by macrophages or to be targeted by complement proteins. Just allows them to get taken out of the system better. So the antibodies do not actually destroy the pathogens, but they are involved in making sure that the pathogens get noticed and taken out by some other members of the immune system. So here's an illustration of this particular response. So up top, this is one where the B cell can respond directly to the antibody, and so the receptor here on the B cell picks up one of these little dots that represents the antigen, and it activates the B cell, and then the B cell goes on to proliferate. It divides many, many times, forming clones of itself. And of those clones, some of them go down and become memory cells, which basically they remain in the blood system. They don't do anything for this particular response, but they're there waiting for the next one, where everybody else then becomes a plasma cell, and they make lots and lots of antibodies that then go out and do their job. This particular picture also shows that there can be a secondary response, which could be even years later, that this antigen appears again, and so then you get plasma cells being made another by the memory cell, and so we've got more memory cells created again, but many, many more plasma cells. So you can see the secondary response is much larger than the primary response, and we'll talk about this later. But this is why you get booster shots. If you get a vaccination, as you're getting subsequent triggers by having the antigen being presented to your body, then it just kind of ramps up your response and becomes stronger and stronger each time. Antibodies are what the immunoglobulins that we heard about, or the globulin proteins. We were talking about the proteins that were present in the blood. We mentioned them. So they are a globular protein that is shaped like a Y. And the two ends of the Y, the top part of the Y here, are where the antigen can bind. So you can see we can kind of clump things together because each one of these Ys can connect to an antigen. Each antibody is specific to one particular antigen, which means our bodies could make millions of different kinds of antibodies if necessary. There's a great variety that is possible. Globular proteins, the antibodies, can be found in fluids all over your body, found in your blood, found in secretions from the mucous membranes, found in your tears, found in the interstitial fluid, which of course is just the plasma that's left the blood and gone in between the tissues. And so, as I said earlier, they serve as identifying labels once they have bound to the antigen on the pathogen. They help activate the complement group of proteins, and by causing inflammation, they help prevent the spread of infection because inflammation kind of allows our body to wall off a portion of that is under attack and keep that infection from spreading. There are 5 different classes of antibodies. You don't need to memorize all these classes unless you're an honors student, but you might hear about them or have heard about them later, before, or we'll hear about them later. IgG, immunoglobulin G, is the most common immunoglobulin. It is the smallest immunoglobulin. It's the one that will go from the mother across the placenta into the developing baby. And so when a child is born, it has a certain amount of immunity that has been given to it by its mother, by the process of the IgG from the mother moving in to the baby's body while it is still developing. It is active against the bacteria, viruses, and various toxins. It is the most abundant and kind of the most active immunoglobulin in our body. IgA is found in those various secretions, the exocrine gland secretions. Getting back to babies, it's the one that's found in breast milk, and again helps to provide immunity, passive immunity to babies while they are still young. The IgA, because it's found in all of the secretions and the mucosal membranes, helps provide protections against the bacteria that would just love to live in that nice, warm, moist environment. And so it's there to keep that from happening. IgM is the type of immunoglobulin that is involved in that blood cell reaction, the blood typing reaction. So it has that role, and it also is the one that is produced first when you have an infection. So the IgM immunoglobulin its production is ramped up first when your body senses that you are under attack. It also helps activate the complement proteins, and it's really the largest of all of our immunoglobulins. IgD is found actually on B cells and is involved in cell activation. And IgE is involved in allergic reactions that helps promote inflammation, which is one of the characteristics of an allergic reaction. Of the immune response, as I already mentioned, you've got a primary response when you are first exposed to a particular antigen. And so the antibodies are being produced as the B cells are coming into play and making more, reproducing and turning into plasma cells and cranking out the antibodies. But it might take 5 to 10 days for a discernible level of antibodies to be detected in the blood. But when your body has developed that immunity, has all of your you've gotten the B cells, the memory cells, all of the machinery in place, then if you are exposed to that antigen again, a secondary response, either the second time or even later, it's all called secondary, but the antibodies will be available, will be at a much higher rate much sooner, and so in only 1 to 2 days you can see the antibodies in the body. So that this next slide, we have a graph. On this graph you can see, the first exposure to the antigen, not only do you have sort of a smaller slope to the line, but you never really get, your peak amount of antibody concentration is only so high. But on your second exposure, you've got a faster response and a much higher response. So this is pretty typical for the antibody-mediated response system, that once the antibody machinery has kind of been put in place and is there in memory, you have a much faster response each time. The same thing is true of your T cells. You've got the memory T cells present in the bloodstream, so they can quickly produce the cytotoxic T cells that can go out there and attack the cells that are infected. Finally, we can think about immunity. This immunity is the ability to resist a particular pathogen, and our bodies, in creating an immunity by the cell-mediated or the antibody-mediated response, are creating an adaptive immunity. They are responding and adapting to a situation. Our adaptive immunity can either be naturally acquired or artificially acquired. So an example of a naturally acquired immunity is this passive antibody that I've already mentioned several times, that happens when a baby is developing. That antibodies move across the placenta from the mother before it is born, so the child is born with a certain amount of immune cells already ready and waiting to protect against any invaders. And then also, if the baby is breastfed, there's additional antibodies that are provided through the breast milk. So this helps keep the child healthy during those first few weeks until their immune system is going to be able to kind of take over the job. And then whenever we respond to a illness, to a bacteria or to a virus, and create the B cells and the T cells, depending upon what it is, and then keep those memory versions, we've then created a active immunity that was naturally acquired. So we have experienced the disease and we have overcome it, and so we now have a natural immunity that is, we made it. It is active. It wasn't given to us by anybody else. But probably and we do active immunity all the time. I mean, the problem with many of our everyday illnesses, colds and viruses and such, is that the bacteria and the viruses that cause a lot of these diseases are reproducing so rapidly that they are mutating, and so they don't come back exactly the same the next time. And so you can have a cold continually because there's about 500 different cold viruses I've read out there. So there's a new one being created frequently as the mutation changes. But the artificially acquired immunity is our whole vaccination system, and it really has been important for improving the overall health of people. In an active situation, this is when you get a vaccine, that the antigens are introduced to the body in a vaccine, but as we saw in our lab, the vaccine is weakened, or it's not quite the same disease, so that our body is able to produce antibodies and the specialized T cells to fight it off, but we are not getting such a live and active and strong dose of the disease that we don't actually get sick for the most part. But we create the machinery to attack the next time that antigen comes into our body system. We're able to defend and defeat it very quickly. And then you can also get passive immunity. I did experience this when I was going overseas, traveling into an area. I got an injection of IgG to just boost the level of antibodies. It was for hepatitis, and so antibodies were taken from somebody who had an immunity to hepatitis, and they were sort of harvested and selected, and I got a selection of IgG antibodies provided to help boost my own immunity. It doesn't last forever because your body will eventually break down those immunoglobulins, but it was enough to cover me for my trip. So if you travel overseas, some of the shots that you get may actually be antibodies, not a vaccination the way we think about it. It's just a sort of a temporary fix and protection that way. So that covers this first part of the immune system. We'll look at some specific immune disorders and finish up the rest of the goals in the next video.